Okay, great. Well, it's 12:05. Welcome, everybody. I'm Richard Kravitz, Director at UC Center Sacramento, and this is the UCCS Speaker Series. Before I get started with the introduction, I just wanted to mention we have some great upcoming Speaker Series events on April 14th, Impacts of COVID-19 on Small Business, and then a week later, a joint presentation from UCLA and Berkeley on science tools and policies needed to predict wildfires, which unfortunately I think we can all bet are coming again this summer. So, if you had asked any of us a year ago whether we would have the number of proven safe and effective vaccines for COVID-19, many of us probably would have laughed. I mean, arguably the COVID-19 vaccine story is a story of some of the most important and rapidly developed medical innovations in human history. And yet there's a lot of questions that many of us still have. And we're really excited to have our speaker today, Susan Buchbinder from UCSF, come here to address them. Some of the big questions that I think Dr. Buchbinder may touch on, or that you may be interested to bring up during question and answer time, are questions about the safety and efficacy of our current array of vaccines, what we can do to get vaccines out there faster, because we really are in a race between vaccines and the new COVID variants that have been emerging. How to deal with this problem of vaccine hesitancy that affects so many different and disparate populations. How to, again, win the race between herd immunity and new variants. How to interpret the onslaught of news stories that are coming out about potential both successes and problems with some of the vaccines, including the new safety data with AstraZeneca, and relatedly, the manufacturing problems that have occurred in the factory in Baltimore with the Johnson & Johnson vaccine. So many really important issues, and I'm sure many more, and we couldn't have anyone more expert to help us deal with some of these than Susan Buchbinder. As I mentioned, she's clinical professor of medicine, epidemiology, and biostatistics at UC San Francisco, where she also directs the HIV prevention research program and serves as COVID-19 vaccine trial coordinator with the San Francisco Department of Public Health. Dr. Buchbinder leads a number of clinical trial groups and has served on several NIH advisory boards. Her work has been published in the journal Science, New England Journal of Medicine, and many more. And she obtained her MD degree from UCSF and trained in internal medicine at San Francisco General. And we're so pleased that she chose to both stick around in Northern California and also is here today with us to tell us what we need to know about COVID vaccines. So Dr. Buchbinder, please.
Thank you so much. So I'm going to be talking about COVID vaccines and take you through from the development to some of the challenges that we have. So just to put this in context about COVID-19, from March 1st of 2020 to January 2nd of 2021, there were nearly 23% more deaths than expected, and that's all deaths, not just from COVID. In previous years, they've only gone up by 2.5% or less in a given year. This is the largest decline in life expectancy that we've seen since World War II, and there are a total of over 500,000 excess deaths from all causes just through January of this year, not even coming up to April, with racial and ethnic disparities with particularly high rates of excess deaths among Black African Americans. COVID was also the third leading cause of death in the United States after heart disease and cancer. And this is from a website, the Johns Hopkins website, that you can go to, and it updates on a daily basis. What you can see is that there have been 131 million cases globally of COVID-19. Over here, you can see that we've had 2.8 plus million deaths globally, and in the United States, over 555,000 deaths, which is really an astonishing number. Unfortunately, California, you'll also see just below that leads in terms of number of deaths in any state in the country. And this down here just shows you the daily cases, and you can see that it looks like there may be another rise that's going on. So this puts the SARS-CoV-2 virus that causes COVID in some context. And what you can see is this is an old slide that started with only 8.3 thousand deaths from COVID. And now we're at nearly 3 million. And so you can see the size of the balls shows you the relative number of people who died from each of these different plagues or pandemics. And we're fast approaching this ball right here, 17th century Great Plagues. But vaccines have been the answer to a number of different diseases. So this is US data only, and it shows you a number of infections in the left-hand column. In the 20th century, in the 1900s, what was the average number of diagnoses per year? And then what's actually happened since we've developed vaccines for all of these diseases. And you can see that there's been, for most of these, over 99% reduction in number of new diagnoses, including smallpox, which is the only disease that we've actually managed to eradicate. And we eradicated that through the use of a vaccine. So what is it that a COVID-19 vaccine could do? Well, it could benefit the individual by reducing the severity of illness or preventing infection. And those are two different things. So we don't yet know whether it prevents infection completely. We know that it reduces the severity of disease, and it could benefit the community if it reduces transmission. But as I said, the only thing we know about the current vaccines is that they reduce the severity of illness. We don't know yet whether or not they prevent infection overall, and we don't know whether or not they reduce transmission. So this is an electron micrograph of the SARS-CoV-2 virus, the virus that causes COVID. And you can see these spikes that stick out that account for its name. It's called a crown or circle. The coronavirus is a crown or circle of light, and that's made up of these spike proteins. And this is a three-dimensional representation of the spike protein in its stabilized prefusion form. What's important about this is that the spike protein is the target of all 6 of the vaccines being tested in the US. There are more vaccines being tested globally and some in early phases in the US, but the ones that the Operation Warp Speed put its money behind, and I'm going to talk about those in a moment. The spike protein is also important for the following reason, and this is a pictorial representation. On the left-hand side is a coronavirus with a spike protein sticking out, and on the right is a human cell with an what's called the ACE2 receptor. And the thing is that the spike protein serves as the kind of key that enters the lock, which is the human ACE2 receptor. And once it enters that lock, it actually opens up the human cell for the coronavirus to enter. So here you can see the spike protein entering the ACE2 receptor, and now it's going to be able to infect this cell. But if we're able to develop antibodies against the spike protein, which is what these vaccines are intended to do, then these Y-shaped antibodies, you can see, interfere with the spike protein docking in the ACE2 receptor, and it prevents infection from happening. So that's theoretically what it is we're trying to do with these vaccines. So let's talk a little bit about the speed with which these vaccines have been developed. And what you can see here is that this is a timeline, and here is when the sequences of SARS-CoV-2 were first published, and it wasn't even called SARS-CoV-2 yet. And here's when the first phase 1 clinical trial started. So from January 10th to March 16th, just 2 months later, we started in clinical trials of a vaccine. That's really unheard of. And it's because there was so much work that had been done on other vaccines that predated this pandemic that were able to just be adapted to be used for this pandemic that we were able to move so quickly. And so the way that we test vaccines is we go through 3 phases of testing. There's phase 1, which is where we're testing safety and tolerability of the product. And usually that involves less than 100 people. We have phase 2, which is larger. It's usually a few hundred to a few thousand people, in which we're looking at the maximum tolerated dose, the best dosing schedule, and looking at the immune response to the vaccine. And then we have phase 3, which is answering the question, does it actually prevent infection or prevent severity of disease? And this one involves thousands of people. And what's important to know is that we're in the testing of each of these vaccines, none of these phases have been skipped, but they have been compressed. And here's what you can see. On the top is traditional vaccine development, where you have a long period of preclinical development in animals before you move into phase 1, phase 2, phase 3. Each of those happens sequentially, often with long pauses between them. Then there's a licensure, and only then do you start large-scale production. On the bottom of this picture is showing you that the phases have been compressed and that they're overlapping. And so phase 1, 2, and 3 have been compressed down. And the most important part is that large-scale production actually began long before we even knew whether or not the vaccines worked. So we took a risk in actually manufacturing, paying for the vaccines to be manufactured before we knew whether or not they would work, just so that once they were, we found that they worked, we would have them ready to be actually administered. So the other way that things have been sped up is that the vaccines that are currently being used in the US are being authorized under an EUA, or emergency use authorization. And that's when, during a public health emergency, the FDA can use its emergency use authorization authority to allow the use of unapproved medical products to diagnose, treat, or prevent serious or life-threatening diseases when certain criteria are met, including that there are no adequate, approved, and available alternatives. So that's what an EUA is. It's not full licensure of the vaccines, but it is just allowing early use of the vaccines while all of the data are being put into that need to be amassed, are put into a licensure application, which takes many months to go through. So for the COVID vaccine, the FDA stated that they would issue an EUA if the vaccine was at least 50% effective, and there were at least 2 months of follow-up after the full vaccination series, and that there were at least a minimum of 5 cases of severe COVID disease in the placebo recipients, so we could see whether or not it actually protects against severe disease. And severe disease is often when you need like high flow oxygen, when other organ systems are involved, when your vital signs are showing that you are in distress. So this comes from the New York Times, and you can go onto the New York Times coronavirus vaccine tracker. It gets updated every few days. This is from April 3rd. And what you can see is the number of vaccines in the various phases, in phase 1, phase 2, phase 3. There are 6 vaccines that have been authorized for earlier limited use, like the EUAs, and 7 that have been approved for full use in other countries. We don't have any that have been approved that have gone through all of the many months that it takes to do the licensure application for these vaccines. Those are still underway, but we do have these EUA vaccines. And then some vaccines proved not to be successful, and so they were abandoned. We have 83 vaccines in these various clinical trials, and as I said, some are in multiple phases of clinical testing because there's overlap and compression of these various phases. And there's 77 more vaccines in preclinical evaluation, so in animal studies before they've even entered human studies. So you can see that we have a large pipeline of additional vaccines, and I'm going to just be going over a small fraction of those here. So we'll talk about the ones that have been tested for in the US. And in particular, I want to say Operation Warp Speed decided that they needed a diverse portfolio of vaccines because they didn't know which ones were going to work. And so what they said is, we're putting our money down on these 6 vaccines, and we'll pay in advance to manufacture these and to get these tested to be sure that they're actually, to see whether or not they're effective. There are 2 messenger RNA vaccines, the Moderna and the Pfizer-BioNTech vaccines. And I'm going to describe how those work in just a moment. The adenoviral vector vaccines, AstraZeneca and Janssen, or Johnson & Johnson's vaccine, And then the recombinant protein and adjuvant vaccines made by Novavax and Sanofi. And those include an adjuvant, which is a chemical that is sort of perks up the immune system and strengthens the immune response. Now, the advantage to messenger RNA is that there's rapid manufacturing facilitating efficient move to the clinic. And that's how we got into the clinic within 2 months. There had already been a lot of work done on messenger RNA vaccines of other types, but they were able to then quickly manufacture once they had the sequence of SARS-CoV-2 to develop a messenger RNA vaccine. So we have 2 of those. The adenovirus vectors, again, also have rapid manufacturing, so that facilitates efficient move to the clinic. And the good thing about the adenovirus is that it has been used before for a licensed vaccine that's been approved in Europe. And then we have adjuvanted recombinant proteins. They're not as fast to manufacture, but they are scalable. And there's several vaccines that we currently use that use this particular approach. So let's talk about the messenger RNA vaccines first, because those are the 2 that have the first emergency use authorization in the US. And what messenger RNA does is it's the template on which proteins are built in our cells. And what they did was they took messenger RNA for just the spike protein. And because it's fragile and we need to get it inside the cell, they packaged it in a lipid coating. That's what makes the vaccine. The vaccine gets vaccinated, the messenger RNA gets kind of dumped into the cell, and then the cell starts creating these spike proteins. Now it's just the spike protein, so it can't, there's no live virus, there's no way that this can form a viral particle, so you can't get infected from the vaccine, but it creates these spike proteins. And what that does is it trains the immune system to see those spike proteins and attack them with antibodies and with cells as well. So that in the future, if you're exposed and have these spike proteins on a SARS-CoV-2 virus in the body, the antibodies will attack immediately and hopefully prevent infection from occurring. So the 2 messenger RNA vaccines are the Pfizer vaccine and the Moderna vaccine. They're very similar, so I've just put them side by side. Both of them use this prefusion spike protein. The Pfizer vaccine gives 2 doses 21 days apart. Moderna gives 2 doses 28 days apart. There's nothing magical about those amounts. Those are just the way that the clinical trials were structured. So that's how we're administering them. The efficacy was around 95% for both of them. And you can see that there were a large number of cases of symptomatic disease in both of the vaccine trials, with very few infections in the vaccine group. In terms of severe disease, there were 9 cases of severe disease in the placebo arm in the Pfizer vaccine study, and only 1 in the vaccine group, and 30 cases of severe disease in the placebo group in the Moderna vaccine, and none in the vaccine group. So it's both of these are very highly protective against severe disease. And that's really what we want to prevent. We want to prevent people from getting hospitalized, needing oxygen, and certainly from dying from COVID. There are no differences in efficacy by age, so it works equally well in older populations, which is great news. And with both of these, there are increased symptoms with the second dose. So I'm going to show you that next. This is the Pfizer vaccine side effects. So people have heard about these. You can see it's not everybody that gets these, but you can see also that these are the systemic side effects: fatigue, headache, muscle pain, chills, joint pain, fever. Some of these, like fever, is fairly uncommon. Others, like fatigue, is fairly common. And you can see that the proportion that get any of these symptoms goes up with dose 2. This is for the 16 to 55-year-olds. What you'll also see is that older people have fewer side effects from the vaccine. So that's one of the advantages to being older, is that there are fewer side effects to all of the vaccines that are currently being tested. I wanted to just show you the Pfizer and Moderna head-to-head in terms of their symptoms. And the way we can do that is that they were collected in the same way by this VSafe program. It's a CDC program that you can sign up for when you get your vaccine. And it sends you a text message on a daily basis for the first week, and then it spreads out how frequently you get texted and asks you about side effects. So of the people who responded, and these were 1.9 million people who responded who had at least who had both doses of the vaccine. You can see that with Pfizer and Moderna, that local pain is the most common symptom, and that Moderna has a slightly higher rate of side effects than Pfizer, but still within the same ballpark. And the reason probably for that is that Pfizer has a 30 microgram dose, Moderna has a 100 microgram dose. So again, higher rates of side effects after dose 2 than after dose 1 for both of the vaccines. And this just shows you about the duration of the side effects. So day 0 is the day of vaccination. And as you go from left to right, these bars stand for injection site pain, fatigue, headache, muscle aches, and chills. And what you can see is that the symptoms peak on the day after vaccination, and then they recover pretty quickly, so that by day 3, most of the side effects have gone away. A little less than 1 in 5 still have a sore arm, and that the peak is higher and may last for slightly longer after the second dose. And this is in people less than age 65. And in people 65 and older, again, many fewer have side effects, and they resolve very quickly. For all people, for all ages, they generally resolve relatively quickly within the first few days after vaccination. So there was a lot in the news a while ago about anaphylaxis, which is a severe allergic reaction, a life-threatening allergic reaction. And it had been seen in 50 cases, this is by January 18th, 50 cases in nearly 10 million people who'd gotten the Pfizer vaccine. That's a 5 per million doses administered, and 2.8 per million doses administered of the Moderna vaccine. And just to put that in context, 1 in 59 people diagnosed with COVID in the United States will die of it. 1 in 59. 1 in 2 of every 400,000 people will have anaphylaxis, and none have died. So there's no question that the vaccine is much safer than getting COVID. So how is it that the adenoviral vectors work? And this is just showing you the chimp adenovirus, but so the chimp adenovirus is the AstraZeneca trial, the AstraZeneca vaccine. The Johnson & Johnson vaccine uses an adenovirus type 26. Both of them are cold viruses. The AstraZeneca one is a cold that infects chimpanzees, but not humans. The Johnson & Johnson one is a human cold virus. They've been weakened so that they don't replicate in human cells. And now, instead of putting just the messenger RNA into the cells, what's done is the spike gene is inserted into the genes of the carrier virus. That virus then serves as the packaging for carrying information about the spike protein into the cell. The DNA then gets converted into messenger RNA that again makes the spike proteins, that again creates these antibodies that will attack the SARS-CoV-2 virus if you are later infected. So what do we know about these adenoviral vector vaccines? Well, we have Johnson & Johnson and AstraZeneca. The vector for Johnson & Johnson is human adenovirus type 26. AstraZeneca's is the chimpanzee virus. The insert is the prefusion spike. That's a stabilized spike protein in its configuration that it looks like before it actually attaches to the cell. AstraZeneca is the only one of the 6 Operation Warp Speed vaccines that did not stabilize the spike protein, but it's still also the spike protein. Johnson & Johnson just has 1 dose. AstraZeneca also has 2 doses. You can see that there was good protection with Johnson & Johnson, 67% overall, but 74% in the United States against symptomatic disease, any symptoms, and AstraZeneca was about 76%. And against severe disease, it was 85% for Johnson & Johnson and 100% protection against death and 100% protection against severe disease in the AstraZeneca trial. So both very highly effective against very severe disease. And you can see the numbers of infections in the vaccine versus the placebo arms in terms of severe diseases. Again, both of them seem to work in the older age range as well as the younger age range. And that's important because the immune system does decline as you age. And so you might expect that the vaccines might be less efficacious in an older population. And the older populations are the ones who are most susceptible to severe disease. And so that's great news that all 4 of these vaccines now have shown similar efficacy in older age populations. And unlike the messenger RNA vaccines, the adenovirus vaccines and AstraZeneca, which has 2 doses, the second dose has fewer side effects rather than more side effects. I mentioned that there are 2 protein-based vaccines. What those do is they just take the spike protein itself and inject that along with the adjuvant. So rather than having the cells make the protein, you just insert the protein itself. Again, it's just the spike component of the virus. It was almost 90% effective against symptomatic disease in a study in the UK. And again, there were 10 cases in vaccine recipients and 96 cases in placebo recipients. It was 100% effective against severe disease in the UK study, with 5 cases overall, all in the placebo group. And we're awaiting data from the US trial, which is currently ongoing. So Novavax does not yet have an EUA. It does not have emergency use authorization yet. We're waiting for the data from the US trial. So let's talk briefly about vaccines tested outside of the United States. And I'm just showing you a subset because, as you saw, there's this huge pipeline of vaccines. But people have heard a lot, perhaps, about the Sputnik 5 vaccine. It's a combined Ad26 and then adenovirus type 5 vaccine. So first you get a dose of Ad26 with the spike protein in it, then you get Ad5. It uses the spike protein, 2 doses, 21 days apart, 91.1% efficacious against symptomatic disease, 100% against severe disease, and it needs to be frozen in terms of storage. The Sinovac, Sinopharm, and CanSino Bio are 3 other vaccines manufactured in China. They've been tested outside of China because China actually had such a good reduction in the number of new infections in their country that they couldn't actually test the vaccines there. So they've tested them around the world. And the thing I want you to notice here is just these 2 are inactivated vaccines. So they've been chemically inactivated so that they can't cause infection. But rather than using the spike protein, they're using the whole virus. There are 2 doses of each. We've got some good indication of symptomatic disease, efficacy against symptomatic disease, but we don't actually have any published data, so we don't really know. And then the CanSino Bio is an Ad5 vaccine against the spike protein. And again, we don't have published data, but this is also a single dose vaccine. So I'm going to turn to talking about 7 different challenges that we face with our current vaccine situation. The first is shipping and storage, and this got a lot of attention early on because the Pfizer vaccine was needed to be stored initially at -80 degrees, which is an ultra cold freezer that very many places did not have. And so it made for a more difficult shipping and storage. Now it can be stored in a regular freezer for up to 6 months, and it's stable at refrigerated temperatures for 5 days. And the Moderna vaccine, similarly, a regular freezer and stable for up to 30 days at a refrigerated temperature. The reason that this is important is that there are, again, places that we need to ship vaccine to that may not have access to freezers or, and certainly the ultra cold freezers. So globally, the AstraZeneca adenovirus vaccine and the Janssen or Johnson & Johnson Ad26 vaccine only need to be refrigerated. Vaccine distribution has been another issue for us. So we're now up to more than 4 million doses a day in the last few days. We've hit that mark. So that's really great. There's been this huge scale up of administering vaccine, which has been really heartening. And so the question is, when will all adults be vaccinated in the US? If the pace continues as it currently is operating, all adults could be vaccinated by the end of June, which is great. That's 78% of the population, because it doesn't include the under 18 age group, and only the Pfizer vaccine is licensed for children, for teenagers 16 to 18. All of the others are 18 and above, but none of the 3 vaccines with an EUA authorization, the Johnson & Johnson, the Pfizer, or the Moderna, are yet have an EUA for use in teenagers under the age of 16 or children. So this is vaccine distribution by state, with the darker colors indicating better coverage. You can see overall there's 31% of the overall adult population has gotten at least 1 shot. 18% have been fully vaccinated, and 78% of the doses that have been delivered have actually been administered. 161 million shots given. In California, we're at almost 33% have received at least 1 dose, and 17.3% have been fully vaccinated. So a little bit better than average in terms of getting the first shots in. So the third challenge is vaccine hesitancy. And we have both sort of some deep-seated vaccine hesitancy or with an anti-vax movement. But we also have COVID-specific anti-vaccine sentiment that we need to contend with. So the Kaiser Family Foundation runs a survey every month in which they ask the public, do you want to get a vaccine, and a COVID vaccine, and when? The answers are either they already got it, or as soon as possible, I want to wait and see, only if it's required, or definitely not. So now we're going to look at the already got, or as soon as possible. So the group that is eager to get the vaccine, and what you can see is that the numbers increase with increasing age. That's probably both motivation, but also the rollout of vaccine has happened in an older age population. You can see that for Black Americans, the proportions are lower than for white and Hispanics. There's a big political divide. So Republicans have the lowest rate of wanting to get the vaccine. Fewer than half said that they want to get it as soon as possible or have already gotten it, compared to independents and Democrats. And again, we have some difference in terms of the rural and suburban population compared to an urban population. If you flip that around and now ask who definitely doesn't want to get a vaccine, overall that's 13%. It varies somewhat by age. It varies somewhat by race, ethnicity, with white individuals being the largest group that definitely doesn't want to get the vaccine. Again, Republicans definitely don't want to get the vaccine. It's the highest proportion, and in rural areas. So we do have a lot of work ahead of us to try to understand why people don't want to get the vaccine and hopefully make it available to them. But vaccine enthusiasm is growing in Black and Latinx adults. So here on the left are Black adults, on the right are Hispanic adults. And what you can see is this is the group that wants to get it as soon as possible or has already gotten it. And that's going up in both groups, whereas this lighter blue line is going down. And that's the group that wanted to wait and see. So fewer people want to wait and see, and more people want to get it as soon as possible. Challenge number 4 is, when is it that children are going to get vaccinated? Currently, the Pfizer vaccine is the only one that's licensed for 16 plus years. The other 2 are just for 18 plus. But Pfizer just this week released data on a trial of over 2,000 12 to 15-year-olds. They showed that they had 100% efficacy against symptomatic disease. There were 18 cases of symptomatic disease. All of them were in the placebo arm. They have a robust antibody response with at least as high as the 16 to 25-year-olds, So that's good news. And it's as well tolerated as in the 16 to 25-year-olds. So it appears to be both safe and well tolerated. And they're going to be applying for an emergency use authorization designation as soon as possible for this younger age group. And several of the companies are also testing their vaccines in children down to 6 months of age, but we're unlikely to have results for those until later in the year. So the fifth challenge is viral variants. And on the left, we see the coronavirus in the center. That's a pictorial representation. This is the RNA inside. And what they've done is just stretched out the RNA on the outside so that you can see what it looks like. And this is the RNA for the spike protein. What we're particularly concerned about are mutations in the spike protein, because those are what might affect the vaccine effectiveness. Each SARS-CoV-2 viral particle has nearly 30,000 letters of RNA that tell the infected cell how to make more copies of the virus. Occasionally, when the virus is replicating, errors are made, and these are called mutations. And a group of viruses that share the same mutation are called a variant. So just to put this into context, these are different viruses: hepatitis C, influenza, flu, HIV, and you can see that the size of the ball represents how diverse the genetic sequences are from person to person. You can see that they're very high for some of our viruses. SARS-CoV-2 is this little speck here that's been blown up 100-fold so that you can see it, but it's actually not a very highly mutationally diverse virus. However, what we're seeing are variants that are affecting the spike protein, and those are of great concern. So there are variants from the UK, from South Africa, and Brazil in particular that we've been tracking. Those are called variants of concern. It appears that the messenger RNA vaccines should be protective against the UK variant, but they may have reduced efficacy against the South African variant. There's a 6 to 8-fold reduction in neutralizing antibody titers, but the mRNA levels are still supposed to, the antibody levels from the messenger RNA vaccines are supposed to still be high enough for protection, but there's limited clinical data so far. Other vaccines have reduced efficacy against the South African variant, so we know that both the J&J, the Johnson & Johnson, and the Novavax vaccines that were tested in South Africa had lower efficacy in South Africa than they did in other parts of the world where there wasn't this B1351 variant. AstraZeneca actually didn't show any efficacy in a small South African trial. And so South Africa was all ready to start vaccinating people with the AstraZeneca vaccine, and they decided to stop and not use it because it didn't show any efficacy. And almost all of the South African infections are being caused now by the South African variant. And the manufacturers are working on developing new versions that incorporate these mutations that could be given as a booster dose if needed. So the sixth challenge is global distribution, and it's really critical. You can see the darker colors are where we have more vaccine, and you can see that where we have more vaccine is in just a few places in the world. There are vast swaths of the world that have no vaccines or very low rates of vaccination. So there was an interesting article in New England Journal of Medicine this week that just talked about from vaccine nationalism to vaccine equity. It's estimated that 80% of the population in low resource settings will not have access to a COVID vaccine this year. This uncoordinated patchwork of immunity could exacerbate the rise of escape variants that could alter vaccine effectiveness. So we're not safe until everyone's safe. And that means that everyone needs to be vaccinated globally. The COVAX is the COVID-19 Vaccines Global Access Program, and it plans to vaccinate at least 20% of the population of participating countries by the end of 2021. And this article was actually calling for waiving pharmaceutical patent protections to increase the production of vaccines because this is this emergency pandemic. So the seventh challenge, can we achieve herd immunity? And it seems that we have promised them herd immunity, sir. And so We don't want to be angry because we don't achieve herd immunity. So what is herd immunity? Well, if only a few people are vaccinated, and they're shown as the blue people here, then one person who's infected, shown as the orange person here, can spread the virus to a lot of other people, because they spread it to one person who then spreads it to a few more people, and so on, until you have a big outbreak in that community. But if lots of people are vaccinated, again with the blue people here, then there isn't much, if they can't transmit the virus, then they will not transmit the virus onward. And this person can only infect another unvaccinated person with whom they come into contact. All of these other unvaccinated people are still protected because they're not coming into contact directly with the individual who's infected. So what herd immunity is it's actually protecting an entire community, including the people who are not vaccinated, if you get enough of the people who are vaccinated, get enough of the community to be vaccinated. And if the vaccine prevents onward transmission. So here's an optimistic view of herd immunity, also coming from the New York Times. And what you can see is that total immunity is a combination of people being fully vaccinated and people having immunity because they've become infected with COVID. And what you can see is that by August, we could achieve herd immunity if vaccinations continue on track, and if you only need 70% protection of the population, and if there is no onward transmission. But there are 5 challenges to achieving herd immunity. First, it's estimated that you need 70 to 90% of the population vaccinated or immune to achieve herd immunity, but we don't yet know whether the current vaccines prevent transmission. There's an important study that just launched in colleges that's actually going to be studying this very question. Vaccine rollout is uneven. In youth, until youth are immunized, you need to achieve 100% vaccination rate in adults before you can reach the herd immunity levels in the US. Globally, we're nowhere near achieving herd immunity levels, and geographic pockets could allow for outbreaks within communities. We don't yet know the impact of variants. We don't yet know how durable the immune response is. And we don't know if vaccines are going to change behavior, because on the way to achieving herd immunity, if people loosen up on non-pharmacologic interventions like masking and social distancing, we'll get more spread. So this just shows you the disparities in distribution. This is, we probably need to get above 70% to achieve herd immunity. And you can see Israel, the United Kingdom, the US, Serbia are the 4 countries with the highest rates of vaccination, and very few beyond that. So there is some encouraging news to end with. There's real world effectiveness in Texas. They showed over 90% effectiveness of staff isolation or quarantine. In Israel, they showed over 90% protection against symptomatic disease and severe disease in real world environment, in which they compared people who have been infected, vaccinated, and people who haven't, including against the B117 UK variant. And in healthcare workers, 80% effective after the first dose and 90% effective after the second dose, again, in a real world setting. And then in terms of durability, there is good news that came out this week that we have 6-month dose post-vaccination data now, and it shows that the Pfizer vaccine is more than 90% effective against severe disease up to 6 months after vaccination. And also there are small numbers, but it looks like the vaccine was also effective against the South African variant, the one that we're very concerned about. But we've also just launched a Moderna vaccine trial in South Africa to be able to test that. So to summarize, we have 3 EUA vaccines in the US. There may be more soon. We could have enough vaccine for all adults in the US by summer. Teenagers will have access soon, younger children later in the year, but we need to address vaccine hesitancy. Real world efficacy is very high and may be durable, but we'll have more data on vaccine effects on transmission later this year. We need to track variants and assess their impact on vaccine efficacy, and booster doses are possible in the future. and we have to work towards global access of vaccines. And with that, I'll take questions and stop sharing my screen. Thank you.
Great, thank you so much for a fantastic talk. And with that, we will be opening it up to Q&A. If folks can go ahead and raise their hand, and actually before we even do that, I'll give Brooke just a minute here to go ahead and stop the recording.